I now have the pleasure of introducing the next speaker, Alexandra Scotti McLaren. Scotti was, she graduated from CNLM in 2021 with the undergraduate BSc in Nutritional Science and the Personalised Nutrition Practice Diploma. She's currently doing an MSc at King's College London in psychology and neuroscience of mental health. Upon graduation at the end of next year, she will open a nutrition practice in Geneva, specialising in menopause and mental health. Her paper, which says is published on the schedule, is actually still pending, but her paper is titled "An Investigation of the Role of Estrogen on the Serotonergic System, Targeting Soy Isoflavones as a Potential Strategy to Reduce Perimenopausal and Postmenopausal Depression." Scotti, hello.
Hello, everyone. Thank you for the introduction. So, in essence, this is a paper on the effects of estrogen and serotonin as a mechanism and looking at soy isoflavones as a potential intervention. So just to introduce it, it is looking at peri and postmenopausal women who have depressive symptoms, looking at soy isoflavones as an intervention. And then looking at the effects of estrogen on serotonin via 5 mechanisms. So those are tryptophan hydroxylase 2, monoamine oxidase A, serotonin transporter, and serotonin receptors 1A and 2A. And then the outcome would be a reduction in depressive symptoms in peri and postmenopausal women, according to a standardized mood scale such as the HAM-D or the HADS. So the scope of the research, it was a mechanistic review. There were 1,421 papers screened, 63 were selected based on predefined inclusion and exclusion criteria, with 2 research aims. The first was the mechanistic search, so to clarify the mechanisms underlying the protective influence of estrogen on the serotonin pathway with the intersection of peri and postmenopausal depression. And then with the research hypothesis of estrogen increasing the production and availability of serotonin through its effects on 5 mechanistic pathways. Again, TPH2, MAO-A, SERT, serotonin receptors 1A and 2A. And then the intervention aim was to evaluate the effectiveness of a clinical intervention utilizing soy isoflavones before and after menopause. So this research was deemed relevant because depression is the leading cause of disability worldwide, affecting over 300 million people. And estrogen receptors are plentiful throughout the brain regions involved in mood regulation, including the hippocampus, hypothalamus, and RAF nucleus. And so by binding to the receptor, estradiol regulates gene expression that moderates neurotransmission of serotonin, which is implicated in depression. And cross-sectional studies suggest that nearly 70% of menopausal depression occurs during perimenopause, although the research regarding the underlying mechanisms is conflicting. And the risk of major depressive disorder has been found to increase 14-fold during the 24 months surrounding menopause. And of course, there are other factors which influence the risk of depression in the menopausal transition, such as vasomotor symptoms, surgically induced or early menopause, and midlife psychosocial factors, such as care for an elderly parent. So here we have the mechanistic diagram. On the right is the classical pathway of estrogen, and on the left is the serotonin pathway. So on the right, the purple triangles are estrogen with their receptors below, and estrogen diffuses into target cells, binding primarily to beta receptors in the brain and forming dimers. And these estrogen dimers then initiate gene transcription in 2 ways. One is by binding to estrogen response elements, or EREs, in of DNA in the nucleus or the mitochondria. So TPH2, TPH2 and SERT are ERE-mediated molecules. And the second way is by attaching to other transcription factors which lack EREs. So this would include MAO-A and serotonin receptors 1A and 2A. And then on the left, we have the axon, the neuronal axon, the synaptic cleft, and the dendrite. So serotonin is in green, the receptors are the green boxes, and estrogen and receptors again in purple. So the blue boxes represent the 5 mechanistic research hypotheses, which predicted the effects of estrogen on serotonin. And the purple boxes predict the effects of estrogen withdrawal, suggesting the effects of menopause. And the lavender boxes then would indicate whether the effects of soy isoflavones on serotonin are similar to that of estrogen. So the 5 hypotheses then state that the following mechanisms are enhanced by estrogen, thus increasing serotonin production or availability. So in the axon, first would be the conversion of tryptophan to serotonin via TPH2. Secondly, the degradation of serotonin by MAO-A. And third, the reuptake of SERT, reducing serotonin in the synaptic cleft. Then in the synaptic cleft, we have the serotonin receptors. So the fourth hypothesis, since 1A is inhibitory, would say that it's further inhibited by estrogen. And as 2A is excitatory, then that would be further enhanced again by estrogen. So these then are our 5 research hypotheses. Also of soy isoflavones as an intervention, which were then researched via systematic mechanistic review. So here we have the results of the mechanisms. So there were 24 animal studies accepted, both in vivo and in vitro, and the available research supported the first 2 research hypotheses, that estrogen increases serotonin synthesis and availability through the stimulation of TPH2 and decreased degradation by MAO. But there was less scientific agreement on the effects of estrogen on SERT and serotonin receptors 1A and 2A, which were the hypotheses 3, 4, and 5. So the results of 6 studies were inconclusive regarding the effect of estrogen on SERT, but results varied according to the method of ovaryectomy and also the influence on the different brain regions. However, a comparison of findings was limited by heterogeneity of the study designs. And then on serotonin 1A receptor, the 4 animal studies diverged in the direction of effect of estrogen withdrawal and treatment on 1A receptor binding and the G protein coupling, and also on the brain region tested. But overall, the results supported the hypothesis in those parts of the brain which affect mood regulation, such as the dorsal raphe. 2 human studies demonstrated limited effect of estrogen treatment on 1A receptor binding, but long-term use of exogenous estrogen by participants may have blunted this effect. And then finally, on the serotonin 2A receptor, again, the results were conflicting. So 2 animal studies supported the hypothesis that estrogen withdrawal decreased 2A expression while estrogen treatment restored it. But there was 1 animal study that found no effect of estrogen on 2A expression. And 1 human cross-sectional study found that binding decreased with age regardless of estrogen concentrations. So these conflicting results may be due to research limitations. First, that the use of young animals might not be representative of perimenopausal age. Second, that the method of estrogen withdrawal might have been incompatible with menopause. The studies tended to focus primarily on pathways other than specifically estrogen and serotonin. Sample sizes were small, and there's always the uncertainty if animal studies are relevant to humans. So then moving on to the interventions, I looked at 26 studies were selected. All of them were human except for 1 animal study. So the availability of studies was quite limited on the effect of soy isoflavones for menopausal depressive symptoms, and the quality of human studies was quite variable. So then the findings were pretty well matched half and half. 9 out of 16 studies found statistically significant effects of soy isoflavones, but 7 out of 19 did not. And the results of the systematic reviews were pretty generally unfavorable towards the effect. But again, we had some pretty serious study limitations. First, treatment, there was varying dosage and duration, which may have affected effect size and significance of isoflavones. Formulations were inconsistent or not reported. Then many studies focused on cognition or menopausal symptoms other than mood, with mood assessed only as a secondary outcome. There was limited inclusion of women with depressive symptoms at baseline. Most studies lacked inclusion criteria of sufficient severity of depressive symptoms or frequency of episodes. Most of the human studies selected had small sample sizes and were likely insufficiently powered. And only one study tested the participants' ability to produce equal, which would have determined the effectiveness of some formulations of soy isoflavones. There was an underrepresentation of peri and early postmenopausal women, and many studies included women of all ages, including reproductive years. And then finally, the mood scales. So among 16 studies, there were 15 different mood instruments used. And this variation would have limited the comparison of depression scores before and after treatment. And the scores were based on self-assessment, so thus subject to inconsistencies among participants and subjectivity. Then looking at the clinical application of soy isoflavones. So again, the research is conflicted on the effectiveness in reducing depression before and after menopause. But again, there were many study limitations, as just mentioned, so that should be taken into account. It is generally a safe intervention approach. There are contraindications for high doses for women who have a history of breast cancer or thyroid issues, but food sources seem to be safe. It's a reasonable approach given the findings on the mechanistic hypotheses. In terms of dosages, one systematic review found that a daily dosage of 50 milligrams for at least 12 weeks was effective. Then it's worth noting this critical window of opportunity for intervention in peri and early postmenopause. And linked with that then, also it's interesting to note that depression is associated with estrogen fluctuation in perimenopause, rather than the low and absolute levels seen after menopause. So this might explain that critical window of opportunity. But it's worth mentioning that discussion was quite absent from the literature. And then finally, this mechanistic review identified some pretty notable research gaps and raised questions meriting further preclinical and clinical studies. Rather than going through all of these, I'd like to just bring your attention to some of the first points. First, that depreciation is associated with estrogen fluctuation rather than the absolute levels in menopause. So then the question remains, why is the risk of depression greater in perimenopause versus menopause? Then there's this critical window of opportunity around the menopausal transition with reduced sensitivity to estrogen with age. So the question is mechanistically, why is that? And then larger studies are needed, which would involve both perimenopausal and early postmenopausal women with inclusion criteria of significant enough depressive symptoms. And I'll conclude with that and turn it back over then to Michelle. Thank you for your attention.